everyone, welcome back to my class. This is Rumana Ali. I hope you all are doing well. In today's class, I'm going to explain you about the technique that are used for our DNA technology. So here, the first technique that we are going to discuss is gel electrophoresis. This is a technique or a process by which the DNA fragments can get separated, and it contains two parts. The first thing is separation of the DNA fragments that takes place by using gel on density gradient, and the other part is about using isoelectric point. First, we'll see how the separation of the DNA fragments takes place by using the gel plate. Here, the first thing that we need to do is the isolation of DNA, and we have already discussed in our previous classes that the isolation of DNA or the DNA which is a gene of interest that can be isolated from the different organisms by using the various enzymes. That means by using various enzymes we can easily isolate the gene of interest or the DNA. After the isolation, we are going to separate the fragments of DNA that is present in the mixture. So let us see how the separation of the fragments of DNA takes place. Here, this is a mixture that contains DNA strand in it. All the DNA strands are present in this mixture. Now, to separate these DNA fragments from this mixture, we are going to pour a solution or chilled ethanol solution. This chilled ethanol is a solution. When we pour this solution in this test tube, the precipitation of the DNA takes place. The DNA strands, after pouring the chilled ethanol, here the chilled ethanol is present, the DNA gets precipitated in this form, and here is the mixture. So in this way, the DNA strands gets accumulated or it gets precipitated in between the chilled ethanol and that mixture. Now this separated fragments of the DNA can be taken out with the help of spatula, and this separation or the isolation of the DNA which gets precipitated in this mixture can be done by using spatula, and this process is termed as spooling. After the spooling, after the isolation of the DNA strand that gets precipitated or that gets separated from the mixture, these DNA strands is going to be poured on a gel plate, and we need to make a gel plate. So here we are using agarose gel. This agarose gel is mixed with distilled water, so it's become a little bit slurry, and that slurry is going to pour on the plate. Here is the agarose gel plate which is ready for separating the DNA. Now the main point is separation of DNA fragments based on the density gradient. In this step, what we are going to do is, we will take this gel plate, we'll keep one trough over this gel plate, and in this trough we are going to pour buffered solution, and in this buffered solution the DNA fragments will also get mixed. When we pour DNA fragments in this buffered solution, along with the buffered solution, these DNA fragments will also move on this gel plate. And one point that we need to remember here is, bigger DNA fragments will move shorter distance, or small size DNA fragments will move longer distance on this gel plate. So this is how the separation of the DNA fragments takes place on the agarose gel plate. So let us see the process of separating DNA by using isoelectric point. Isoelectric point is nothing but it's a pH where the particles loses its charge. Now in this method, we will take one trough. This trough shows the presence of negative electrodes and positive electrodes, and here we are going to take one pH scale as well. So this is the pH scale, and in this trough medium is taken. In this trough a gel slab is taken whose consistency is like jelly. Now if we keep the DNA fragment here, then it loses its charge at isoelectric point. So the different DNA fragments shows the different isoelectric point. Isoelectric point is nothing but it's a pH value where the particle or where the DNA fragment loses its charge. We know that DNA particle is always negatively charged. If we keep a loop of mixture which contains DNA here, then the DNA from this mixture loses its charge. For example, it loses its charge at pH 5. This DNA shows the isoelectric point 5. In this way, the different DNA strands shows the different isoelectric point, or it shows the different pH where it loses its charge. Wherever they loses the charge, that pH value will become their isoelectric point. In such a way, we are separating the DNA strands in a horizontal line. After getting separated, this gel slab is taken out. It is going to be placed on the agarose gel plate, and again the buffer solution is taken in a trough, and along with the buffer, the DNA fragments again go, or the DNA fragments run along with the buffer, and the bigger molecules move shorter distances, and the shorter molecules of the DNA fragments, or the shorter strands of the DNA fragments, will move longer distance. In such a way, the DNA fragments are separated on a gel plate, that is agarose gel plate. After the separation of the DNA takes place, now if we want to detect that how many DNA fragments are separated or at which distance they are present on this agarose gel. Agarose gel is translucent, and even the DNA strands will also be translucent. So, it is a little bit complicated to identify the DNA strands that are present on, or that runs on the agarose gel plate. Now, to identify the DNA strands, we are going to expose this gel plate to the fluorescent light. So, when it is exposed to the light, these DNA fragments appears orange colored line. In such a way, the light can be used to detect the DNA strands that are present on the gel plate. Before exposing to the fluorescent light, or before exposing this gel plate to the fluorescent light, first we need to dip this agarose gel plate in a solution called ethidium bromide solution. After dipping the gel plate in the ethidium bromide solution only, we can detect the presence of the strands on the gel plate by exposing it to the fluorescent light. When we expose this gel plate to the light, then the DNA strands that are present on the gel plate, they will appear as an orange colored line. In this way, gel electrophoresis is a process by which the separation of the DNA fragments can be done by using the agarose gel plate, and it can be detected or identified by using the light, by exposure of the gel plate to the light. Gel electrophoresis is a process by which the different fragments of the DNA get separated. Depending upon the particles that are getting separated by this process, gel electrophoresis, we will see the different terms that can be used. So, if the DNA fragments are getting separated by using the process gel electrophoresis, then the term southern blotting can be used. Or if the pieces that are separated by gel electrophoresis are of RNA, that means if RNA is getting separated by using the process gel electrophoresis, then the term northern blotting can be used. And if the proteins are getting separated by gel electrophoresis, then that process can be termed as western blotting. So, depending upon the particles or the pieces of DNA, RNA or proteins that are getting separated by the process called gel electrophoresis, we can use the different terms. If that particles or the pieces that are separated by the gel electrophoresis are DNA, then that process is called as southern blotting. If they are RNA, then it is northern blotting, and if they are proteins, then this process can be termed as western blotting. So, after the exposure of the gel plate to the UV radiations, we can easily visualize the orange color line that are nothing but the fragments of or the strands of DNA, and that DNA can be extracted along with the gel. Extracting or isolating the DNA strands from the agarose gel plate can be termed as elution. So, after the separation of the DNA fragments, that DNA strands or the gene of interest can be inserted into a vector. So, our second step is insertion of isolated gene into a vector. This is a foreign DNA that has been isolated and separated. This foreign DNA is a gene of interest or a desired gene that we require. This is a plasmid that acts as a vector Vector is nothing but gene carrier. So it carries this desired gene or the foreign gene into the host. So what happens? Let us see. The first thing is the restriction enzymes. These are the enzymes that cut both foreign DNA and the plasmid DNA at specific points. So after cutting the foreign DNA and the DNA of the plasmid or the DNA of the vector at specific points, so one specific enzyme called ligases, they helps in attaching this foreign DNA with that of the vector DNA. After the attachment of the foreign DNA with that of the vector DNA, now we are having our DNA. This DNA is called as chimeric DNA or rDNA. rDNA is going to be transformed into the E. coli cells, and when this E. coli cell undergoes division, then rDNA will also get multiplied. So this is about the step called insertion of isolated gene into a vector. How we can insert a desired gene or a gene of interest into a vector, and that vector helps in carrying the gene of interest into the host cell. Let us discuss about the process called PCR. PCR stands for polymerase chain reaction. This takes place for the amplification of DNA. So what do you understand by the term amplification? Amplification is nothing but division. So how the gene of interest gets divided? The gene of interest can be divided or it can be amplified by the process called polymerase chain reaction, and this is a process which consists of three main steps that are: first one is denaturation of DNA or the denaturation of gene of interest. The second one is annealing, and the third one is extension of primer. Let us see what happens in the first step. Denaturation of DNA or denaturation of gene strand means we know that it contains two strands. Gene of interest contains two strands. It's a double stranded one. So it will be showing 5' to 3' end of one strand and 3' to 5' end of another strand. Now the denaturation means the breakdown of the hydrogen bond so that it would get separated. These two strands would get separated, and in the denaturation process, the high temperature, these DNA strand, the double stranded DNA is going to be exposed to the high temperature. It is going to be treated in the high temperature. That means it is going to be heated at a very high temperature so that the denature or the denaturation of the gene of interest can take place, but the temperature should be properly maintained so that the nucleotides would not get disturbed. Only the hydrogen bonds should be broken down in the denaturation of gene of interest. After the breakdown of the hydrogen bonds, these two strands would get separated. This is one strand and here will be the other strand. After denaturation process, the second step is annealing. In the process of annealing, we are going to add primers to these strands, and the primers should be anti-parallel. Primers are nothing but RNA. RNA strands are going to be added in the second step, that is called as annealing. So anti-parallel RNA strands should be added. So here if it is 5' to 3' strand, then in this strand we will take a primer that shows 3' to 5' end, and the next one is 3' to 5' strand of DNA. So here we will take the strand of RNA which shows 5' to 3' end. In such a way, anti-parallel RNA or the primers needs to be added, and that process is called as annealing. Now after the process of annealing, in the third step, we will take DNA polymerase enzyme and deoxynucleotide. So in the third step, that is in the extension of primers, the deoxynucleotides are going to be added and a complete DNA strand can be produced. So in this way, extension of primer leads to the formation of DNA strand. And here, as we are taking the enzyme DNA polymerase, this DNA polymerase helps in the extension of the primer. And as this is a process that takes place at a higher temperature, the enzyme DNA polymerase which are present in our body, that enzymes cannot withstand this much high temperature. So we need to isolate these DNA polymerase enzyme from the thermostable organisms, the organisms that lives at a very high or extreme temperatures. From that organisms we can take or we can isolate this DNA polymerase. So we will be isolating this DNA polymerase from Thermus aquaticus, which is a thermostable bacteria. The DNA polymerase which is isolated from this Thermus aquaticus is called as Taq polymerase, and this is thermostable one which can withstand higher temperature. This Taq polymerase is helping in the extension of primer, that is in the addition of nucleotides. In such a way the nucleotides are being added up and we can get 2 DNA strands. In the first step we have seen that only one strand is present, and this one strand undergoes a process called polymerase chain reaction that gives 2 strands. In this way amplification of DNA can take place by the process called polymerase chain reaction. Polymerase chain reaction contains 3 main steps. First is denaturation of DNA, that is nothing but exposing the DNA strand or treating the DNA strand at a very high temperature so that the DNA strands would get separated. After the DNA strands separated, here in the second step primer is added, and addition of anti-parallel RNA, that is nothing but primer. So addition of anti-parallel primer is called as annealing, and after the annealing extension of primer takes place in which nucleotides are being added up by the RNA primer, and after the addition of the nucleotides it results in the formation of 2 new DNA strands in the presence of an enzyme called Taq polymerase. So this is about a process called polymerase chain reaction. That's all for today. Meet you next class. Thank you.